Okay, I want to welcome you to Lecture 4. This is involving bacterial genetics and biotechnology. Now, for some of you might be wondering, what does bacterial genetics have anything to do with microbiology? Well, you're going to find over time that understanding bacterial genetics, A, led us to understand how bacteria was developing antibiotic resistance. B, how some bacterial genetics principles were able to demonstrate for us how some strains of bacteria were able to pick up other unique proteins or exotoxins, endotoxins, even though they were completely different species. Also, the principles of bacterial genetics helped us to develop the techniques and tools for biotechnology. Now, again, biotechnology for some people might be very far removed, but really it's not. As a matter of fact, a lot of the developments from biotechnology have yielded new drugs, new foods, new flavorings, new processes to purify and make better foods and plastics and biological materials, new crops. But also we've been able to develop very sophisticated tools for diagnostics and tools that now we are able to start beginning to cure individuals of genetic disorders. So we're going to go through all of that as time goes on. So first off, I encourage you to take time to review page 204, both for the key terms and for the glimpse at history. You need to get down the vocabulary for these sections to help you to clearly understand things. Now here you have a bacterial cell that's been ruptured, and really what is happening is it's spilling out its entire chromosome. Remember that bacteria are haploid. They only have one single set of genes. Mostly it's on one circular chromosome. So any change to the genes will quickly show up in a change to the bacteria. That's where we get into these terms like genotype, phenotype. Genotype is the genetic makeup of the organism. Phenotype is the observable characteristics displayed by the organism. Since there's only one copy of the DNA, any change to DNA will show up in the phenotype, the observable characteristics. Offspring of these changes will also exhibit these changes. There's where we come into the term mutation. This is an alteration in the DNA. Now, organisms that have changed sequence or arrangement of nucleotides in its DNA are referred to as mutant. Bacteria can acquire genes from other bacteria via gene transfer. And then we have to come up with the concept natural selection. This is the selection by the environment of cells that are best able to grow in that environment. So think about it from just the simple process of saying, I have a bacterial infection, I'm going to take an antibiotic. The antibiotic goes into the environment, your body, and it now selects against those bacteria that are susceptible to that antibiotic. But some bacteria that may be resistant because they have various mechanisms now are selected for thriving and spreading within that same environment. We'll get into this later. When we talk about wild type, this is a strain of an organism whose properties are similar to the organism isolated from nature. So basically, it is not mutated, it's not altered, nothing. And also remember that not all mutations, changes from the wild type, lead to observable changes. If you remember that we talked about the genetic code a couple of chapters back, we talked about the principle where you can have a degenerative code. So in other words, if one of the codon letters changes, it may not yield a change in the observable consequence, which would be a change in the amino acid. No change in the amino acid, no change in the functional protein. I hope that makes it clear. Also keep in mind that mutations may result in changes to the start or stop codon. The best way I usually give this is with an analogy. I'm going to give you a car, but the problem is at the assembly plant, the car was not 7 or 10 feet long. They just kept building onto it and building onto it. So now the car is 50 feet or 100 feet long. Well, nobody can drive a 50-foot long car, especially in some of the tight turns and corners, etc. in Boston. It's worthless. But we kept assembling the car and adding on to the metal and adding on to the body without recognizing the start or stop sequence. In this case, the stop sequence. This is going to influence the expression of the gene and may alter the length of the gene product. Sometimes mutations have led to extensions of the protein, and those extensions of the protein, far longer than the natural one, the normal one, render that protein useless. And a lot of times you're going to have this happen in situations of frame shifts, which we will explain. Now, a prototrope, that's cells that grow in the absence of any added growth factors. I think I mentioned this last time. A really good microorganism will be able to thrive in a medium that is very, very chemically basic. Water, some sugar, some chemical salts, and that's it. It's able to assemble all of those items it needs. But what happens is that you're going to get from time to time organisms that lose the ability to synthesize a lot of their basic components, certain proteins, certain vitamins, certain lipids, and as a result, they have to live in specialized media that supplies that, okay? And this is where you get into an auxotroph. This is an increase, oxo, mutant cells that require an organic growth factor. So the growth factor might be a vitamin, it might be a particular amino acid, whatever. Now, horizontal gene transfer. DNA transferred from one bacteria to another via conjugation. Think about horizontal as you have two bacteria side each other, and what happens is that one bacteria sends over to its neighbor a little bridge of cytoplasm, encased in, of course, cell membrane. And that in that cytoplasmic bridge, it is going to pass over some genetic information. Now, this is a process that I just mentioned that's referred to as conjugation, but we have other means which are called DNA-mediated transformation and transduction. We'll get into more of this in a little bit. Now, we have to talk about some of the processes here of mutation. This is to get you started, because everybody always asks, well, how come the medicine doesn't work anymore? Well, think about this first. Start off with the idea that you have cells that grow at an incredibly fast rate. Take E. coli. Remember I said this before. It goes through binary fission every 20 minutes. So basically, in an hour, you've gone through 3 binary divisions. You go through 24 hours, you've got 72 divisions. So you could literally start off with one cell and end up with n to the 72nd power minus 1, which is well over 1 billion cells. Now, you have to keep in mind this other thing. In those billions and billions of cells that are reproducing, you're going to have some opportunities or situations that are just going to cause a spontaneous mutation. That is a genetic change that's going to result from the normal cell process. Something was misread, but the misread little error has caused a change in the DNA, and that change is going to pass on to subsequent generations, okay? Now, the rate of mutation, and that is the probability that a mutation will be observed in a given gene each time the cell divides, that rate is generally expressed in an exponential form. So in bacteria, it's usually 10 to the minus 4th up to 10 to the minus 12th. To help you somewhat, think about it this way. 10 to the third is 1,000. 10 to the minus third is 1,000ths. So 10 to the minus 4th is 1/10,000. So what I'm saying in another way is this. If you have a bacteria mutation rate of 10 to the minus 4th, that means that 1 out of every 10,000 might have a mutation. Does that help? Now, this can get into more incredibly large numbers too, but here's another thing to think about. Well, 1 out of 10,000 doesn't sound like much. Yeah, but what if you're making billions of cells a day? Then you're going to have and generate various mutations, and it's going to be passed down to subsequent generations. And so you start increasing the variability as you increase the rate of reproduction of individual cells. Now, mutations are stable. They rarely are going to revert or correct themselves and basically revert back to wild type. Reversion is an event where the mutated gene changes back to the normal state. Okay, and you can see on the top here, you've seen the mutation. You see down here the plasma transfer rate, and that's what's referred to as vertical gene transfer from one generation to the next. You want to keep in mind all of these different formats. Now, horizontal gene transfer is going to have, when you have one generation passing by another, either related or unrelated, and it passes over some genetic information via a plasmid. Okay? And as you can see here, this is a pretty basic understanding of things, but let's take a look at this from another perspective here. And that is at the base rate. Remember that we talked about DNA having being made up of nucleotides. The nucleotides have nitrogen-containing bases, A's, T's, C's, and G's. Okay, so as I'm going through DNA replication, you can see there about midway, you've got A's with T's, C's with G's. But what happens is that if you have an incorrect nucleotide incorporated, you're going to have a base substitution, and that's in the yellow. That's instead of it being AAG, it's AGG. And AGG creates a new strain of the bacteria with the mutant. The next time that the DNA is separated, that G will now force the other strand to become a C. The wild type has an AT, the mutant has a GC, and this may alter whatever the protein product is. There are different types of mutations caused by this incorrect nucleotide base being incorporated. Now, by the way, if it only occurs on one base, one single teeny weeny base, it's called a point mutation. And there are various types of mutations. For example, there's a missense mutation. That's a change in the amino acid sequence due to that actual mutation. A leaky mutation, this is a mutation that renders the gene product only partly functional. So think about it this way. If I had a mutation in hemoglobin, the hemoglobin still works but doesn't carry oxygen as efficiently or completely. That would be something like a leaky mutation. A nonsense mutation is a mutation that changes the codon to a stop, meaning that literally the protein is made, but it's abruptly cut off. Think about it from that car analogy I gave you. Oh, I'm going to give you a car, but instead they only went about halfway. So I've got the front part of the car, the engine, and the hood, and two wheels, and then about right after the driver's seat, it's missing. Well, I can't drive a half a car. I got to have a full car. Same thing. You can't have half a protein to do the job. It has to be complete. Now, a null, otherwise known as a knockout mutation, the mutation totally inactivates the gene. There's no express product, okay? And then we have to deal with this other concept called frame shifts. Frame shifts. Oh, wow, what happens there? Well, you can see some of the mutations here in this particular figure. Frame shifts is where actually the starting of the reading is altered, and you can alter it by starting either one or two of the bases in advance. A mutation that shifts the reading frame of the codons. Now remember, if you affect one of the codons and you keep shifting, this is why they call it a frame shift. If you, instead of reading it at the proper site, let's say the arginine, but you start reading it one codon, one base different, it's going to cascade and affect the rest of the continued codons. So it tends to generate new stop codons. The gene product is either short or abnormally long. The frame shifts will generate new amino acid sequences. Okay? Now, transposable elements. This is also referred to as jumping genes, and people get into this and go, what do you mean? You mean it's alive? It jumps around? This is where we get into Barbara McClintock's Nobel Prize-winning work. A transposon is a special segment of DNA that moves spontaneously from one site to the other in the same or in a different DNA molecule. So think about it this way. It's as if you're listening to a piece of music and suddenly in the midst of it, let's say you were taking classical music, a little Mozart, and suddenly in the midst of it, you suddenly have this rap pop in. And then after about 30 seconds, it pops out and you go back to your Mozart. Well, that's strange. How could that happen? Well, this is what happens when you have these pieces of DNA. They suddenly insert themselves into other genes. They may disrupt the actual function of the gene. They may change the entire gene product. And the transposition is the process of transposons moving to different DNA segments. Now, any gene that the transposon enters into usually is going to have its functional product disrupted. I want you to be aware of that. Here's the situation. Barbara McClintock was studying why there were colored kernels. And she eventually found that since the color is determined by a gene product, some of the genes were jumping in and out, altering the color within the individual kernels, not just each other kernel. As you can see there's some brilliant reds, but it's not completely a red kernel. It's sort of flailed, and then some parts of it are yellow, some parts of it are white, etc. It almost looks like candy than it does actual kernels of corn. By the way, there's a micro assessment at that point on page 208. Take some time to review it. Now, we've been talking about what happens naturally in nature. Scientists after World War II really began to start ramping up their research in biology on mutations and DNA and RNA. And they started saying, you know what, let's see if we can induce some of these mutations. Now, if you sit there and say, oh, that's baloney, why would you want to do that? Well, let me tell you something. In the history of the development of penicillin, one strain of penicillin producing mold was actually produced by just exposing it to X-rays. And then they started to notice that the chemical penicillin was slightly different. And therefore, they had a new type of penicillin that led them to use that commercially. And it was not radioactive or anything else like that, but it was just by causing some changes using X-rays to the fungi that was making that penicillin. Okay. Now, since the natural means of mutations is low, the other way to study mutations is basically create them by inducing them with various agents. A mutagen is a substance that produces mutations in DNA. Now, you're going to see this in table 8.1, a variety of them, chemical and radiation. And you see some of the actions and some of the results. Keep in mind that you don't have to memorize everything, but you do have to start getting familiarized with some of these. And it's been these studies also that have led us to develop tests to determine if, whether it's a food product or a preservative or a pesticide or an herbicide or certain therapeutic drugs, whether they actually can cause mutations and therefore could be considered carcinogenic or mutagenic or teratogenic. Teratogenic meaning it affects the development of fetuses. Okay. So let's take a look at first chemical agents, all right? And the first batch that we're going to look at are going to be chemical modifications. And chemical modifications, any chemical treatment that alters the hydrogen bonding properties between base pairs, so between the purines and pyrimidines, between the A's and T's and the C's and G's. And what will happen is you develop, as a result, mutations in the DNA. Some act chemically by modifying the bases already present. One chemical that is very well documented is nitrous acid, not nitric acid, but nitrous acid. Alkylating agents add an alkyl group. This is a short chain of carbons to the bases. Therefore, they alter the hydrogen bonding properties. One clear one that's done there is nitroso-guanidine. Now, base analogs. If you take a look, you'll see this listed. Base analogs are compounds. They resemble structurally the purine and the pyrimidines, close enough to be incorporated into the DNA during DNA replication. These compounds will alter the hydrogen bonding. Remember, A's with T, C's with G's. This has a certain set number of hydrogen bonds to the complementary strand. Therefore, you start disrupting things and it can lead to mutations. An example is 5-bromouracil, okay, and you can see that up on the chart. But you can also do this with other chemicals, 2-aminopurine, for example. Now, intercalating agents are a 3-ring planar molecule that intercalate. In other words, they get actually in between the nucleotides. They push the nucleotides apart and thereby induce mutations during DNA replication. One well-known chemical is known as Atheneum Bromide. Those that are in biotech, those that are in molecular biology know Atheneum Bromide because when you expose it to ultraviolet light, it glows. It gets in between DNA, so when you're doing gel electrophoresis, you add Atheneum Bromide, and it will kind of hang along with the DNA going through the gel, and it will stick there, and it will act as a lantern when you expose the entire gel to DNA, so you know that piece of DNA contains some DNA. Excuse me, that piece of the gel contains DNA. Pardon the error. Transposition, this is insertion mutation. This is the knocking out of the gene by the insertion of a transposon. We've talked about transposon earlier. Now, what about radiation? This is why we get so concerned about radiation, whether it's X-rays, ultraviolet light, or even more so gamma rays. But let me put the two that are best known here. Ultraviolet light. Okay, you know how everyone goes and tells you during the summer, don't go out and get really sunburned. It is not the visible light spectrum that induces damage to the skin and eventually can lead to skin cancer. It is the ultraviolet radiation that comes from the sun that partially gets protected by the atmosphere, but still can come to the surface of the Earth. UV light induces interstrand thymine binding, which leads to base substitutions. Now, usually what happens is this. As you can see in the diagram there, the UV light is going to activate the SOS repair mechanisms, which are a very error-prone method of DNA recording. What happens if you have two thymines next to each other, remember A, C's, T's, and G's? Well, if you have two T's together, that UV light is going to suddenly create covalent bonds. Now, this is different from hydrogen bonds. This will cause a distortion on the DNA strand. You will have chemical bonding between the two thymines, and that will cause, when the DNA is replicated again, an error that may turn out to be no longer when they replicate the strands, there'll be two T's next to each other. It could be an A, a G, a C. It just gets randomized. Now, X-rays are even more intense. That's strong enough to actually break a single strand. And double-strand breakage of DNA, as well as alteration in the bases. Now, if you go from UV light to X-rays, you're increasing the amount of energy in the photons that carry or convey the light, okay? So you go UV, and then a much higher level is X-rays, and the level above that is gamma rays. And gamma rays are far more intense, can also break strands, single strands, double strands of DNA, and alter the bases. That's why ionizing radiation, as it's called, which can create ions of some of the individual atoms that make up the DNA, for example, the bases, etc. That's why it's so important to protect yourself against that, okay? Now, what we have are some mechanisms that repair damaged DNA. The problem is they're not always the most efficient, the most effective, and in some cases, they're really sloppy, almost like Keystone Cops. If you remember the old videos, Keystone Cop, bunch of guys just swinging around, driving around crazy, and they're just not really doing their job really well. Let me give you some examples here, and you can see this also in Table 8.2. Correction of mutations, basically for error repair. It's essential. All long-term survival of organisms require this. It's only when the mutations are proved to be an advantage to the species are mutations helpful. Now, there are good mutations. There are some that are indifferent and silent. There are some that are bad. So you want to keep that in mind. Mutations may also be helpful in the study of the role of select genes in the survival of an organism. Now, mutations are corrected for by the following systems, except to note that the SOS repair is less efficient and appears to be the method of last result for mutation repair. The big key problem with SOS repair is that it tends to also introduce other mutations in the process of repairing the DNA. So what you first have are proofreading that occurs through DNA polymerase enzyme. Keep in mind that DNA polymerase is not merely just something that goes wonk, 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 wonk down and assembles a new strand of DNA, but it will also have a part of the protein structure have the capability to correct errors. It cuts out the mistake after making the DNA strand, and in doing so, the correct nucleotide base is put in. Now, another area is mismatch repair, and you can see this on 810. Mismatch repair, what you have there is an endonuclease. This is an enzyme that basically cuts inside of a strand of DNA. It recognizes hydrogen bonding errors. It kind of walks along the double strand, and if it finds a hydrogen bonding error, let's say there's something that should have been 3 instead of 2, etc. Remember, I believe it was A's and T's have 2 hydrogen bonds, and C's and G's share 3 hydrogen bonds. So if you've got this awkwardness, that could lead to a strand buckle or warping. So this endonuclease comes in, removes a section of the strand with the incorrect nucleotide. DNA polymerase then arrives, fills in the missing nucleotide strand, and what patches together the sugar phosphate strands. And if you notice that the lower part in between 4 and 5, you have the action of another enzyme called DNA ligase. We're going to call that sort of our molecular glue. Keep in mind some of these enzymes are going to be used in biotechnology later. What about repair of a modified nucleobases in DNA? Oh, okay. Well, let's take a look at this. Now, modified nucleobases can sometimes happen. There could be oxidation damage, and basically that can result in base substitutions if it's not repaired prior to DNA replication. So there is an enzyme, glycosylase, which removes the oxidized guanine. In this case, the oxidized guanine you see at number 1. It will pop it out, make a cut, remove basically a small area of the strand. DNA polymerase repairs it, and then everything is working great. And of course, to seal up the gap, you got DNA ligase again. Now, what about thymine dimers? Well, those are a little tricky. Not really. As you can see in the top here, the photoreactivation and the excision repair process. There are really 2 versions of thymine dimer repair. And these 2 versions will correct the thymine dimers created by ultraviolet light. There is the light-based reaction, which is photoreactivation. Think of photo and needing light. And the enzyme uses light in the visible light spectrum. Yep, what you're reading right now, okay, to cleave or cut out the covalent bonds, okay? And once it's done that, everything is great. See there, the double bonds were covalent bonds between the 2 T's, the 2 thymines, and they've been clipped out. Now, there's also a dark excision repair process. Here, you don't need light. There's an enzyme that removes a section of the DNA where the dimer exists. A second enzyme replaces the missing strand, and the DNA ligase attaches the new strand part to the rest of the DNA strand. Okay, so what about this keystone cop stuff that we were talking about? Well, that's where we get into SOS. Now, before we get into assessing mutations and everything else, I wanted you to just listen to this very briefly. It gets very complex at times. The SOS repair system is made up of 20 genes. New DNA polymerase avoids the lesion and synthesizes DNA, but usually this is not an incomplete process. This new DNA polymerase tends to read off the template that it's going to copy and make the repaired DNA, and it gets error prone. And so it usually is the repair system of last resort because it induces many new mutations, okay? All right, now, we're going to talk about mutations, consequences, and advantages, and then selection. When we talk about natural selection, this is the selection by the environment of those cells best able to grow in that environment. Remember I gave you the antibiotic example? Hang in there. Mutations allow unique opportunities to examine how changes in the environment favor or disfavor the shifts in the environment. Okay? Now keep in mind, shifts in the environment can be levels of oxygen dissolved, temperature, going up, going down. It can be acidity or pH, etc. Now, some of these mutations will not enhance the survival of the organism, perhaps until you actually have an environmental shift. Okay. Combination therapy. This is where two or more drugs are administered at the same time to treat diseases that have the presence of strains with drug resistance. Any mutant cells that are resistant to one drug will usually be sensitive to the other drug and therefore be killed off by combinations of two or more different drugs. This is also similar to the principle that we saw with AIDS therapy, where they made basically this protease and other modified nucleotide inhibitors. It's one thing to have some viruses have resistance because they've made mutation against one drug, and other viruses having mutations against another drug. But when you hit them with this so-called cocktail, a mixture of these different products, usually it'll wipe out everything. Okay? Now, this is based, if we talk about bacteria, on that the mutant rate of more than one gene is the product of the rate of individual genes. Thus, the chance of two or more mutations present in the same bacteria becomes very small. We'll get back to that with antibiotic resistance later on in the course when we talk about more about antibiotics. Mutant selection. How can one isolate select for cells that have a specific mutation? In other words, if I have a billion or more cells, how can I sift out and find that one or a couple cells that have that? And isolate the mutations in a large population of bacterial cells. Well, there's two basic approaches. One's direct, one's indirect. Direct. You see this on figure 813. It involves basically inoculating the cells onto a medium that the mutant can grow, but the parent cannot. Remember the parent type, otherwise known as the wild type. The wild type is always the type that does not have a mutation. The example provided and what you see there, 813 selects for a mutation that is resistant to an antibiotic. In this case, it's streptomycin. Okay? The parent's not going to thrive in the presence of that antibiotic. Okay? And so what you end up doing is selecting only for the streptomycin-resistant mutant. With the indirect method, and you'll see this on 814, the process involves isolating auxotrophic mutants that require a growth factor from the parent that doesn't have it. Okay? So what I'm talking about there is basically all the colonies grow with the nutrient agar. With just basic medium agar, which would be glucose and salts. The auxotrophs don't grow. The auxotrophs lack, because of mutants, the capability to take the salts and the glucose and make the essential building blocks to replicate. So they die out. The wild type basically will thrive on glucose and salts, just as it would easily thrive on nutrient agar. Okay? Now, how do I know what's what on those colonies? Well, if you're trying to study a bunch of different colonies, there is a very safe and nice process called replica plating. Now, you may see this also in your lab exercises or maybe a YouTube video, but it basically goes like this. You use sterile velvet, and you replicate the colonies on each plate. You basically take a master plate of bacterial colonies, press it onto the sterile velvet. Now, you got to make sure that you have a certain marker to kind of tell you what's north, south, east, west, or something like that. The colonies are imprinted on the velvet. They will retain that. And then what you do is you take your plates, whether they're glucose, salt, or nutrient, And then you start having these brand new sterile plates come down at the same position, press on the velvet, they'll pick up some of the bacteria cells in each of the different colonies in the entire array or pattern. And that way you can make a bunch of plates that have the same pattern of distribution of the bacteria cells, but you can then select, depending on the type of agar that you have, which ones are going to be able to thrive on that and which ones are not, okay? So this is called indirect selection method, because you're indirectly selecting for the autotrophs. Notice that the autotrophs would be missing. That's the indirect approach. Now, let's also talk about conditional lethal mutants. Oh, excuse me, before I do that, let me talk about penicillin enrichment. That's pretty cool. This process takes advantage of the prototroph's constant growth and auxotroph's failure to grow in the absence of growth factor. Penicillin is added to the auxotroph-prototroph mix on basic media. The antibiotic destroys the prototroph cells, but doesn't affect the auxotrophs. Penicillinase, the enzyme, inactivates the penicillin. Then the autotroph-rich culture is added to the medium that contains the growth factors. What am I talking here? First off, prototrophs can handle it. They're just like wild type. So they're going to be able to just bing, 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 grow like nothing else. But if you add penicillin and you incubate it, all the prototrophs are going to die out. That's because of the presence of penicillin. Auxotrophs will not be able to reproduce at that short period of time, because notice that they are in with the prototrophs the same media, which is glucose salts broth, which basically means auxotrophs will not be able to take off and grow. But what you're doing here is basically killing off all of the prototrophs by the exposure of penicillin. Then you add penicillinase to the nutrient agar that you're going to pour out onto that plate. The penicillinase will break down the penicillin. You've already killed off the prototrophs. All you should have left is auxotrophs. Auxotrophs will grow on the new type of medium that's going to basically be rich in culture products for the growth of auxotrophs. I hope that becomes clear now. Conditional lethal mutants. These are mutants that have mutations that will kill the cell under select conditions. One example of that is temperature sensitivity. Some temperature-sensitive mutants will grow only at lower temperatures, but when your temperature rises, they die. So the colonies that have the temperature-sensitive mutation are favored for growth in lower temperatures. Okay. Now, we're going to get into something very interesting. It's called the Ames Test. This is a well-known test. Yes, there was a gentleman by the name of Ames, and he was out on the West Coast. He was at, I believe it was University of California, who developed this idea. This is basically a test for carcinogenicity or mutagenicity of various compounds using a histidine-requiring auxotroph. Now, remember we talked about auxotrophs? They don't have the capability to do something. Well, in this case, the auxotrophs are what is referred to as his minus. That means that they have an inability to make histidine, an essential amino acid. So, what you do is you're actually looking to see if the chemicals that you're going to add along with the auxotrophs will cause a mutation that would cause a reversal, that the actual organism will be able to grow even without histidine. See, presently, those histidine auxotrophs require histidine in their medium for them to grow, okay? The mutations are selected for what's called revertants. Those are the ones that are going to have a mutation and presumably for the histidine synthesis. But you have to understand this is based on three assumptions. The reversal of the histidine synthesis pathway can be readily measured. The frequency of the reversions can be increased by the mutagens, and most carcinogens are mutagens. Usually, if you have something, like aspartame or olestra or any of the other food additives that are out there, they'll do these tests. Before the FDA gives approval, they'll say, okay, how was the Ames test? Now, the other problem that you have to say is, but you're taking this food product and you're putting it into someone's body. So it's going to get broken down in part by such organs as the liver, etc. So a lot of times they have to follow it up by trials, since the substance could be altered by the metabolism. In other words, it could be not a mutant, but once it gets into the body, converted to a mutagen. So what they do is they take a rat liver extract, and this is added to the bacteria plate. It's going to test for enzymatic activation of the mutagen. And finally, the more accurate method of the test uses a strain of bacteria that lacks its excision repair system, the mutation repair. And therefore, this increases the isolation of any mutants that develop. So this is something where they've added certain processes to enhance the visualization or the coming to surface of any mutants that might possibly occur. Now we're going to start talking about the actual gene transfer between bacteria. Now, besides mutation, genetic information in bacteria cells can be altered if the DNA comes from other cells. Does that mean I could have horse DNA put into me and suddenly I'd have a set of horse legs? Maybe I'd look like a centaur? I don't think it goes that easy. But with bacteria, it's a lot easier. As a matter of fact, horizontal gene transfer, this is called the lateral gene transfer, basically the transfer of genes from one bacteria to another, is in part one of the major reasons that we have antibiotic resistance being such a major threat today. Now, horizontal gene transfer genes from one bacteria to the other. Vertical gene transfer, DNA is passed from parent to daughter cell. So I want you to be aware of that, okay? I'm going to be dealing with some of this in a few minutes. You need to get familiarized with several of the terms here. Recombinants, these are cells that have received DNA from other cells. Homologs, these are identical genes. Homologous recombination, the replacement of a host DNA with foreign DNA of the same genes. So if I take and I put into the gene, into a bacteria cell, the gene for making histidine, and it crosses over with the defective histidine synthesis gene, and now that synthesis gene has now been incorporated into the chromosome of the bacteria, you now have the capability to synthesize histidine, and so the bacteria now can thrive in histidine-absent environment where it could not previously. When we talk about a donor, we're talking about cells that supply or donate the DNA. Recipient is the cell that receives the DNA from the donor cell. A replicon is a DNA that has the origin of replication, ORIs. We're going to come back to that term a lot, so you want to keep that in mind. It's sort of the special site on a DNA that permits DNA replication prior to cell division. So it allows this replication to occur. It can be on the small loops called plasmids, or it can be on the larger chromosome of the bacteria. And this is regardless of whether the fragment of chromosome, basically chromosomes and plasmids are replicons, whereas a fragment of the chromosome is not. So in other words, to help you understand this, if the chromosome, or a smaller loop called a plasmid, has an ORI, they're replicons. But if I just chop a piece of the chromosome out, like you saw spilled out earlier, and it doesn't have an origin of replication present, it's not, that piece of that chromosome is not going to be a replicon. Three mechanisms we're going to talk about. That is transformation, otherwise known as DNA-mediated transformation, transduction, and conjugation. And by the way, there is a review perspective 8.1 on page 219. I encourage you to review that, okay? Now, DNase, by the way, is an abbreviation for deoxyribonuclease, which is an enzyme that degrades DNA. So you want to be aware of that. What we have with DNA-mediated transformation, also called DNA transformation, is a process where the transfer of naked DNA in the environment to the recipient cells. Some of the DNA passes through the cell walls and cell membranes via DNA receptors. These pieces of DNA can then be incorporated into the chromosomal DNA of the recipient cell. Now, keep in mind we talked about the ORI, the non-integrated DNA fragment. And you can see the integrated DNA fragment there, okay? Now, when we say something is competent, this is a condition in which the bacteria cell is capable of importing DNA into its cell and incorporating it into the chromosome. This process of DNA-mediated transformation is very common in many gram-negative and gram-positive cells, as well as S. cerevisiae is known as Saccharomyces cerevisiae. It's basically a type of yeast. If you go to 819, you're going to see this. Now, what you see here is in the perspective, that's Griffiths' work. And Griffiths' work was the first demonstration of genetic transformation. What he did was he took organisms, he injected live encapsulated cells, mouse dyes of this type of ammonia. Now, at the time he called it S-pneumoniae, and I believe we call it today pneumococcal. But here's the basic point. He took living unencapsulated cells and has no effect. Now, this particular strain of bacteria has two different versions, those that have an encapsule and those that don't. He then took heat-killed encapsulated cells. He was trying to actually work on means to treat this because pneumonia is a nasty disease. And he found that it had no effect. But here was something that happened. There was an accident, and they mixed some tubes, and they took heat-killed encapsulated DNA. Heat-killed encapsulated cells, mixed them with living non-encapsulated cells, injected them into the mice. Come back the next day, mice are dead. When they do an autopsy, and they did a microbiological recovery, they found living encapsulated cells isolated. So what happened? Well, the heat-killed encapsulated cells broke and released their DNA. The living non-encapsulated cells accepted those fragments of DNA, which included the capacity to make encapsulated cells. They were converted over to encapsulated cells and were able to then create pneumonia and kill the mice. Here is a better view. As you can see here in this diagram, you have basically a situation of streptomycin capability. I believe streptomycin resistance. Yes. First step you have to have is always entry of the DNA. You'll notice that the double-stranded DNA is rendered single-stranded before it can enter the cell by the nucleases. Then you end up with integration of the donor DNA. You have a breakage and reunion of the homologous sections of the DNA. This is where you're going to integrate the donor DNA into the host chromosome. Last step. You end up now having this new information, new DNA, incorporated into the chromosome. And so all of the cells henceforth, replicating by binary fission, you're going to have the multiplication of the transformed cells, and they are going to basically have that trait that was originally transferred from that donor DNA. Now you can do this a little bit more by force nowadays. We have what's called artificial competence, making the cell competent to receive the foreign DNA. There are different biotechnology techniques. One's called electroporation. Basically, you take the fragments of DNA, You mix them in with your bacteria cells, and you add an electric current. It forces the foreign DNA right into the cells. And usually what happens is the electric current actually creates pores in the cell membrane to permit the entry of the DNA fragments, or you can use much larger pieces of DNA in the form of plasmids. You have to keep in mind that DNA is a negatively charged molecule, therefore it's always going to be repelled by electric current flow. Okay? From there, we need to go into transduction. Transduction is a little bit different, and what that is basically you're going to take a phage. Now, phage is also referred to as bacteriophage. These are unique viruses that attack bacteria only. You can't get bacteriophages that attack eukaryotic cells. They've just never been able to do that. But they are looking at phages, and we'll get into this in a little bit more discussion in the virus chapter, where they've been able to consider using the phages to treat bacterial infections in humans. And in the East Bloc, in the Soviet Union, last century, they were trying to develop that technology. Okay, but let's just get into this now. In transduction, you have a virus, and it contains a small amount in the viral DNA of some foreign DNA. And this is usually from the previous bacteria that the virus infected. So it'll be carried into the next host bacteria cell. The foreign bacterial DNA may not start a new round of viral replication, but rather it becomes incorporated into the new bacterial host cell's DNA. The process is very common in many gram-negative, gram-positive cells. And there are two types, generalized and specialized. Generalized is any gene from the donor cell that can be transferred, whereas specialized, only a few specific genes can be transferred. And you see what happens is that instead of making new viruses, you have the cell altered by transduction, and now passing on that new incorporated DNA that is integrated into its chromosome into the further daughter cells created by binary fission. Okay. Next we got to move into is conjugation. Now with conjugation, and this is interesting, you notice one cell in the other cell here. This is an image you're going to have in figure 8.21. The interesting thing here is this. This is basically an exchange of DNA between a host cell and a recipient cell. This requires physical contact between the two cells. Now, there are these extensions from the cells called pili. Some of them have been referred to as sex pili. Basically, they allow bacteria to come in contact with a recipient bacterial cell. Now, the exchange can use single-stranded DNA from either the chromosome or the plasmid. Now, we need to talk a little bit more about what plasmids are. They are small loops of DNA. They're smaller than the chromosome, and you can have up to like 20 or more. I believe I've mentioned this before. Usually, a functional plasmid will also have an ORI, an origin of replication, because as the cell replicates, it will also replicate both the chromosome, and you will also get copies of the plasmids, and they will also be passed on to the daughter cells. Now, in the plasmid transfer, take a look at 8.22. Here's where you get into the process. Okay? So your transfer of the DNA starts at the site on the plasmid called the origin of transfer. So it begins to sort of unwind a copy of itself. And the origin of transfer, there's a slight cut there, and that single strand passes out through the F pilus bridgeway that's been created. And what happens is the F plasmid, the fertility plasmid as referred to, must be present for the donor to send out the DNA copy. Now, in my notes, it says keep in mind that the copy DNA does not travel through the pili. So it's not going through the pili. The pili allows sort of an attachment and for the cell to bring together itself, and then it forms the cytoplasmic bridge. Now, the cells that are donors that have the F plasmid are referred to as F+. Okay? They're the donor cell. This F characteristic allows for cells to pass the traits on to other cells through the process of conjugation. Usually an F plus cell will come in contact with an F minus cell, all right? And afterwards, the F minus cell becomes F plus. Now, you got to keep in mind something too. The cells can spontaneously lose their plasmid. Those that do are said to be cured. The F plasmid, well, we've got to talk about that a little bit more. The presence of the F plasmid can allow other plasmids to copy and send a copy of their DNA to the recipient. This is as long as the donor cell has an F plasmid, okay? So think about it this way. The F plasmid is an initial infection, and basically it allows a larger transfer of DNA and the traits that are coded on that DNA to the other bacteria. Now, some of this is, you have to understand also, on plasmids can be other genes, not just genes for F, but for antibiotic resistance, for having new particular traits, let's say pili, or for having other metabolic capabilities. Donated DNA is always in the single-stranded form. It's made double-stranded once it gets inside the recipient. And the homologous recombination occurs for foreign DNA to integrate into this recipient's chromosome. So whenever you put this foreign DNA from an F plus to an F minus, usually what happens is the F minus will integrate a lot of this material into its own chromosome, thereby making it almost in essence permanent. F donors. Now, F plasmid can be excised from the bacterial chromosome. When this plasmid leaves, it takes a small piece of the bacterial chromosome with it. And the F plasmid is now referred to as F prime. Now, I hope that hasn't been too harsh on you, because you can see here, F is a major important characteristic. And you need to get this down very well. This is partly how you have a lot of bacteria picking up new and unique traits, etc. And from here, you can see making the contact, transferring the ends, etc. Now, let's talk a little bit more about plasmids and the mobile gene pool. See, what I'm trying to get you to understand is that bacteria are not merely just sort of, they're there. The same bacteria that you're experiencing is the same bacteria that the caveman or Christopher Columbus or dinosaurs dealt with. There's constantly subtle changes that are developing, and in part because the bacteria are being exposed to different environments, and in those environments they may have a leg up in survival or a leg down, okay? When we talk about the core genes, we talk about the conserved genes that make up the species found in all species of the organism. When we talk about mobile gene pool, that also known as the mobilome, these are DNA sequences found in only some strains of given species, and this includes plasmids, transposons, phages, and genomic islands. Okay, here's some examples here. Now, why is this so important? Because some of these carriers can alter some of the strains of bacteria. You may have heard of some individuals, for example, talking about E. coli. Well, that E. coli is okay. T12, we worked with it, yeah, in the lab. The E. coli that, kills people who haven't got have consumed incompletely cooked hamburger, such as the Jack in the Box incident in the 1990s, and they all died. Well, that was E. coli strain 017-H7 and so on and so forth. This is unique strains that start popping up, and in part it is because they picked up from the mobile gene pool the traits capable of causing a lot of damage. Okay, You got to keep in mind also that the extra chromosomal piece of DNA that multiplies independently of the chromosome, that's the plasmid. It's usually a closed loop of double-stranded DNA. And it can contain useful but not vital for survival genes, but the genes can range anywhere from a few genes on that loop to several hundred. And the number of copies of the plasmid can vary from 1 to several hundred. And the traits are very, very different in various different strains. Here are some plasma-coded traits. Antibiotic resistance, lots of them. Pili synthesis, tumor formation in plants. Nitrogen fixation, oil degradation, gas vacuole production, insect toxic synthesis, plant hormone synthesis, antibiotic synthesis, increased virulence, toxin production. So you see what I'm trying to say here is, as you study the genetics of bacteria, now this is a brief part of really studying, I've studied much more, but it gives you some perspective about how come these things are all starting to get harder to treat? Well, either antibiotic resistance or because they have an increased virulence, which means increased capability of causing disease. Okay, now, our plasmids, they confer resistance traits to many different heavy metals and antimicrobials. Many plasmids are composed of two parts: the resistance genes, the R genes, and the resistant factor, transfer factor, which codes for transfer of the plasmid to other bacteria. So what you see in this example alone is the origin of transfer, the pilus synthesis genes, the origin of replication, but here it comes, chloramphenicol resistance gene, carbonicillin resistance gene, and tetracycline resistance gene. Each one of these genes has one or two capabilities that lead to the resistance of the bacteria that has these genes in them. And here's another pointer. Some plasmids can transfer between unrelated species and even between gram-positive and gram-negative. So this is what I'm trying to get you to have an appreciation for. The next thing we're going to deal with is transposable elements. Now, I mentioned to you before transposons. These are mobile genetic elements, and they can move from around the chromosome and shut off gene function wherever the transposon integrates. Like I said, suddenly you're listening to some wonderful music and it shifts completely. What happens? Somebody drops some music in between the recording of the file. Transposons can also be a mobile element that under the correct conditions can move pieces of DNA along with the transposon. So if you get several different transposable elements side by side, suddenly when they take off, they can also carry along something else like antibiotic resistance gene. The structure of a transposable element is as follows. There is an insertion sequence, otherwise known as IS, and you can see that there on the figure 824, which I would just go back to, if you'll give me a second here. 2, 3, 4. There we are. And what you see are some transferring parts. What I'm going to do is go forward here. If you notice the insertion sequences here, okay, and notice how the arrows are pointed inward. What that means is that if both insertion sequences lifted up now and pulled away, and of course the edges, the purple edges on that lower diagram, the composite transposon, come together, they'll be fused together. And now you've got, in essence, what could be a plasmid that contains transposable genes and an antibiotic resistance gene, and it can go other places. Now, IS codes for the enzyme transposase. This is the enzyme that allows for transposon movement. You have a mutation or a damage to those IS segments, you're not going to be able to have that gene jump, okay? The enzyme is flanked on each side by an inverted repeat, which is usually 15 to 25 bases long for the sequence. The enzyme must recognize the inverted repeat to act to remove the transposon from the DNA strand. IS systems can be transported to other cells via plasmids. So now you see where the plasmids are forming. In some cases, the key point that I want you to walk away from is the composite transposon. This is where on each side you have a transposable element, the mobile element. It has totally functional inverted repeats. It's got a totally functional transposase gene. But now what's in between those two insertion sequences could be a serious gene that could cause problems down the road. Okay? Finally, we get into this other area. Oh, and by the way, the perspective 8.2 offers some very interesting aspect. I know some students aren't interested in the plants, but it might give you some idea the next time you see these big lumpy galls on trees, look knobby looking, etc. This is all caused by certain bacteria, but it is also that bacteria that's provided us with tools to eventually develop means for plant genetic engineering to produce fruits and vegetables and plants that are resistant to certain pathogens, etc. And you can see that here. The Agrobacterium tumefaciens gene, it's got a TI plasmid, and you would see this crown gall tumor, this infection in the stem. But when you start taking apart that, you can then isolate that tDNA and start doing some marvelous work to make better disease-resistant plants. Now, I've just got to go a couple more minutes here with just the point of virulence factors and genetic islands. When I talk about virulence, this is the relative ability of a pathogen to overcome body defenses cause disease. When we get to the host pathogen interactions later on in the chapters, we're going to talk a lot more about virulence factors. But think about it this way: to keep from being consumed by a white blood cell, to release certain toxins, to cause tissue necrosis. These would be examples of virulence, okay? And basically, the ability to create toxins and other virulence factors is passed genetically from one strain of bacteria to the other by the above methods. In other words, transposons, conjugation, etc. When I talk about the term genetic islands, genomic islands, these are large DNA segments in a cell's genome that originated in other species. We are seeing more and more of that as we are able to deepen our understanding of the genetic pattern that exists in different strains of bacteria. Not to scare you, but just to understand. As our depth and breadth of knowledge and understanding this occurs, we can see how these species are able to overcome our best medical treatments. Finally, there is the term pathogenicity islands. These are patches of DNA in bacteria. They code for virulence factors. For example, E. coli strains have some islands that favor urinary infections, while there are other E. coli strains that will favor gastrointestinal cell damage. Okay? Now, what are the barriers to how bacteria defend against invading DNA? Well, in short, we're going to get into this a lot more very soon. But basically what we have are the mechanisms that detect foreign DNA once inside the cell by deoxyribonucleases. These are enzymes that degrade foreign DNA. The mechanism also allows for stability of bacterial genomes against invasion of foreign DNA. In other words, wait a minute, you're telling me about how they switch and everything else. How is it that all of them haven't become homogeneously all mixed? The super bacteria. Because they're enzyme systems that are part of what is called the restriction modification system. And we're going to get more into this in a bit when we talk about restriction enzymes. When I talk about F1. Oh, and by the way, I may have jumped a little bit ahead, but we're going to have so much fun in biotechnology. What you got to understand is this. We're going to be talking about restriction enzymes and modification enzymes. And these enzymes will provide you with certain capabilities to cut, okay? To cut out and basically clip out pieces of sequence, and you can make them either with flat ends, in other words, there's nothing sticking out, or you can have what's called cohesive sticky ends. Now, what happens is this. Restriction enzymes restrict the entry of foreign DNA into the cell. As we've been developing research since the 1960s, we've developed more and more restriction enzymes. And some of them are very important because they help to prevent bacteria from being infected by viral DNA. They're a type of the endonuclease enzymes. There are some that do these clips, but they clip only at very specific sites on the DNA that's being brought in. Very unique nucleotides, okay? So the sequence might be GAATTC on one side. They will clip it. Now, if you have just one base pair difference, though, you can't get that particular restriction enzyme to cut it away. We also have modification enzymes. These are enzymes that add methyl groups to adenine and cytosine. The methyl addition does not affect gene expression, but protects the DNA strand from restriction enzymes. And these are all going to be the really neat tools that we're going to be jumping around and going over in Chapter 9. I apologize if it was a little bit ahead, but sometimes there's some material that gets mixed in. I do want to tell you that when you finish Chapter 8, you want to take time to go over the perspective, the microassessment, the future opportunities 8.1, and the chapter summary. When you get into biotechnology, which is 9, which we kind of already did a little, you want to take time to review the glimpse of history as well as the key terms. The key terms are going to be extremely important. And we're going to bounce through them, and I'll try to give you some introduction. But it's very important that you understand these, because here's the other thing. You're going to be dealing with some of this both on the lecture side as well as on the lab side. So let's talk about what the definition of biotechnology is. The use of microbiological and biochemical techniques to solve practical problems and produce more useful products. That's basically it. No, it's not to make the X-Men. No, it's not to make monsters. No, it's not to make your worst nightmares come true. That's all Hollywood hogwash, okay? When we talk about recombinant DNA techniques, these are techniques used to create new combinations of genes within organisms. Genetic engineering, this is the process of deliberately altering an organism's genetic information. Restriction enzymes, these are the molecular scissors, I talked about them earlier, for DNA. They will selectively cut the DNA strand at specific sequence sites. These are the restriction sites. When I talk about gene cloning, otherwise known as DNA cloning, this is the process by which genes are inserted into a replicon, such as a plasmid or a virus, which is then inserted into a cell where the replicon can replicate. Okay, so keep in mind, replicon, it's got to have something what? Origin of replication. It's got to have this and that. The replicon is that piece of DNA that can replicate. It could be large, it could be small, but it has to also have the origin of replication. Otherwise, it's just going to be floating around there as a meaningless piece of DNA. You want it passed down into the next generation of bacteria or yeast cells or whatever. Okay? We've talked about restriction enzymes. Oh, and by the way, there's a wonderful table. You get the terms for the applications of the DNA technology. Here are some common restriction enzymes. Now notice where the arrows indicate the cleavage sites. Notice the recognition sequence. Okay? So let me go over a couple of other pointers here just to help you before we start jumping all over the place. And yes, I have a degree also in biotechnology studies, and it's really interesting to see how things have moved from the 60s and 70s and 80s now to the 90s, 2000, and the 2000 teens. So many different things are going on now, and so many improvements to life and health, as well as technology and other means because of biotechnology. Okay? What you see here on this table is how certain enzymes, and they tell you the source. You see, all the enzymes don't come from one particular bacteria or another. They're actually found in a variety of both prokaryotes and in some cases eukaryotic life forms. How they get these strange letters? Well, basically what happens is the first or second letter is capitalized. And the first couple letters indicate what species it came from, microbial source. Then what you get is either a number or series of numbers or letters that indicate. So if you wanted to have ECO 1, you'll find Escherichia coli enzyme 1. ECO 2 was the second one found. ECO 3 was the third one found. There are whole directories now. And some of them are very specific. They will make unique cuts in areas of about 6 pairs of nucleotide bases. Others may jump around, so you get bigger cuts. You get smaller cuts, bigger cuts. And what I mean by that is they'll chop up the DNA because this particular sequence is very common, whereas other sequences are very, very rare. And so you get bigger pieces of DNA. Okay? Now, the cloning vector, this is a piece of DNA that acts as a carrier of a copied fragment of DNA. The insert is the DNA that's been incorporated into the vector. Now, before I go too wild, I want to give you this idea. If you take a look up here, you have your bacteria. Then you cut up a bunch of pieces of it with restriction enzymes. Then you find a vector, which would be some type of, for example, a plasmid. You cut it, clip it at a particular site so that it's linear. Then you join it to your fragments. Then you take this now cloning vector and insert it into bacteria. Once inserted into the new bacteria, you can research it. You can do DNA production to make more sequences of this, or you can see that this new DNA would make certain types of proteins, vaccines, or proteins for industry. Okay. By the way, where do you get a certain type of dyed genes? Is they have a process where they take the newly made genes and expose them to an enzyme that will tend to bleach out some of the indigo dye to give you that stone wash genes effects, and it hasn't been put in with stones. It will remove some of the dye, but it also handles high heat because it was obtained from a archaea type of bacteria that lives in high temperatures. Oh, yes, and by the way, back here, you could also, how am I sure that the gene fusion has occurred? Well, you can put in a gene that also encodes for a readily observable phenotype. The bacteria colony may turn green or blue or white, or it may fluoresce. In other words, give off some bioluminescence. When we talk about transgenic organisms, these are organisms that have foreign DNA introduced into the genome. And if you go back just one, you can see some of the products made. And you can see this, transgenic plants, DNA for study, a variety of proteins, whether it's chymosin for cheese making, bovine somatropin, which is for increasing milk production in cows, the vaccines of hepatitis B, HPV, and foot and mouth disease, a variety of pharmaceutical products, alpha interferons, gamma interferons, growth hormone, insulin. That's how we now have humalin. That's human insulin. They took out the insulin gene and put it into a particular culture cell, and we're able to produce it. Tissue plasminogen activator, TPA. So all of these are products from genetic engineering and from doing this process. Okay, I'm just going to move very briefly back on a few points. Restriction enzymes, which we've talked a little bit about. And the different enzymes are obtained from different bacteria. That you know. And I've talked about the naming process. They're called restriction enzymes because they restrict the entry of foreign DNA into the cell. That's when they were first discovered in the 60s. The unique DNA degrading enzymes will cut the DNA strands at specific sites These pieces will make it available, and then later on, other enzymes will come and chop up the rest of those smaller fragments. The site where the restriction enzyme cleaves is called the restriction sequence, which consists of a specific sequence 4 to 8 nucleotides long, and this has been exceedingly useful in genetic engineering. Some other endonucleases are less select, will degrade any nucleotides and cut the DNA into single nucleotide pieces. Okay, now the two things you got to be aware of is this. You take a look at the ALUL enzyme. It will create what are referred to, because of where the cuts are, they're edge on edge, the CG and the GC. So you'll end up with what's called blunt ends. There's no overhanging amount. Whereas if you looked at the BAM-HI, it will create an overhang, okay, or they'll call these sticky ends. So you end up with one piece of DNA having this extension, GATC, and the other fragment that gets cut away having a GATC on the other piece. And so what happens is, if you want to, you use BAM-HI for your vector, and you create the same type of sticky ends, and then you can basically, with DNA ligase, fuse all of it together, and it makes a nice cloning vector, okay? The next thing to go to is gel electrophoresis. And by the way, I do include some videos, and I encourage you to take time to look those over. It'll help you understand this. First off, remember, DNA is a negatively charged molecule, okay? And we have to separate out these restriction fragments of DNA. You know, you want to get big pieces, small pieces. You don't want to just have a jumble of DNA in there. So what you do is you use an electric charge to force the DNA through an agarose gel. Now, agarose, although it looks solid, is almost like this huge sponge with big holes, small holes, etc. So the smaller fragments will move much faster through the gel than the bigger fragments. And what you can do to tell what you have, if you notice there's something there called size standard. These are commercially available pieces of DNA that are of different lengths. Kilobases, KB, means thousands of bases. So you've got some that are only like 2,000 bases long, others that are 23,000 bases long. Okay, KB is 1,000 base pairs. So you put the size standard in one of the rows, your samples in the other rows, and then you run the electricity going from negative to positive, which is going to push the DNA through. And what you will end up with is a gel that has a bunch of different bands of different sized DNAs, and you can make an understanding of how big these are. And as you've separated them out, you will be able to then visualize them using special stains, and then you'll be able to extract the DNA from the pieces of gel. Yeah, you can use little X-Acto knives and cut out the little block and then extract it out, and then you can do other things with that piece of DNA. Okay, now we're going to talk about first prokaryotic genes and eukaryotic genes. And there's a little bit of difference, and it's important. Okay, first off, when you isolate the DNA from bacteria, the cells must be lysed to break them open and extract the DNA. Piece of cake. Much of the broken DNA pieces are then broken into smaller fragments via restriction enzymes, then inserted into plasmids. Plasmids are recombinant DNA molecules. They're inserted into bacteria cells, and they multiply these fragments in the population of new transgenic bacteria cells. Oh, okay, that makes sense. So basically, I can go back here, take my cuts, then go here, and chop up the DNA, put them with vectors, put them into new bacteria. The bacteria reproduce, and they'll make copies of the cloned vector as well, and I can then do all sorts of studies with it. Yes, that's true. But since you're going to have a bunch of these different fragments of different sizes and different genes, etc., you may form what's called a library. It is a collection of cloned molecules of the genome of the organism of interest. So therefore, in that library, you may have to then separate out what's actually going on. If you take a peek here on page 239, that's exactly what you do. Each library will contain different pieces of that recombinant molecule and thereby have different genetic traits. Now, this is in the beginning what they would do. Later on, we began to analyze these fragments and we're able to put them back together again using computer programming to get the basic map of the entire genetic blueprint, shall we say, for bacteria X, which you see listed there. Now, what about eukaryotic? Well, what do you mean? Eukaryotic, a little bit trickier. Remember we talked about how eukaryotic DNA, the DNA would make a copy in a messenger RNA, but it hasn't left the nucleus yet. And that pre-messenger RNA has to cut out the introns. And the introns are the useless coding material. The exons are the useful. And then they have proteins that will fuse or splice together the exons. And then you have this mature, ready to go messenger RNA that gets exported outside of the nucleus to be then taking that message information and converted into a protein. Okay, sounds good. But does that happen in a test tube? No. Sorry, doesn't always happen. What you can do, though, is you can isolate the messenger RNA from the cytoplasm, and then you can study what we call the cDNA, the copy DNA. In other words, think about it this way. If I just take messenger RNA that's been isolated, use an enzyme called reverse transcriptase, and that takes and makes the DNA from the messenger RNA, I'm going to get a strand of cDNA. Yeah. And that's without the introns. Yeah. And that would be a functional protein. Yeah. And so the DNA polymerase would be able to complete the cDNA because you go from one strand basically to two. And then what you have to do is you can insert that into a cloning vector. You might have to do some trimming on the ends. It might have blunt ends. It might have cohesive sticky ends. And then you put it into whatever you want. You want to put it into a eukaryotic cell in culture or to a bacterial cell, and then you would get your product. Okay? Now remember that there is the annealing part, which is also referred to as the paste. And of course, your paste would be DNA ligase, which would be the enzyme that helps to re-anneal or actually secure the foreign DNA with the vector, the plasmid DNA, okay? Usually, when we start talking about vectors, it's usually a plasmid. There are some specialized, larger expression vectors, and there's actually bacterial artificial chromosomes. In other words, what I'm trying to say to you is, see, what happens is you have this capability, an ORI, you have this capability to make this protein from this DNA, so the gene product, but sometimes you've got a much, much bigger piece of DNA that you want to insert. This is where you get into the larger structures, and they call them bacterial artificial chromosomes. They've also done this for yeast. Now, why do we have yeast as a consideration? Because yeast is eukaryotic, and therefore it has the potential, not always works, but the potential to clip out introns. So if in that big piece of DNA you inserted, you could still be able to study it afterwards, and you'd be able to study also the introns removed. You have to keep in mind something too. Steps to host selection depend on the gene products made and whether the host cell makes harmful toxins like endotoxins. That's part of the trickiness. You see, I can't make certain drugs if I know that I'm going to have to destroy the bacteria cells, and the cells themselves make nasty little endotoxins that human beings would be sensitive at the nanogram or femtogram range. That's a big no-no. Inject that into me and I go into shock. That would be a bad day. It would also be the end of my company if I was doing this for biotech. Transformation. Well, the transformation process would be electroporation. Transformation selection, usually what you're going to do is some sort of antibiotic resistance or reporter genes. And by the way, I forgot to show you the reporter gene. This is a cool one that really happened in several research projects that occurred. One of the coolest ones was one that seems to be sort of a picture you'll see a lot of. If you just look for fluorescent plant, in the late 80s, researchers were able to put into a tobacco plant, and the reason they use tobacco is because it's well studied as a plant, and they were able to put in the fluorescent gene for the tobacco plant. So all they needed to do, and this gene is called luciferase, and they poured luciferin, which was a liquid, which is the precursor and causes the glowing effect once it's in contact with luciferase. And you could literally see the entire, from the roots going all the way up through the stem into the leaves, the plant glow. Now that's the effect. The luciferase is the reporter gene. Now we have other green fluorescent reporter genes found. Some of these have been extracted from jellyfish, etc. And this is where you're going to have this conformation occurring. Some other ones use certain coloring colonies. You might see this, for example, if you go to 9.9, okay? The bacteria cells plated on the medium containing this chemical called XGAL. And XGAL, when you have a functional LAC-Z gene inserted, makes the colonies appear blue, okay? Now, a couple of other pointers here to help you. When we have large fragments of DNA, and we're much more complex, and you're going to get that with eukaryotics, aren't you? I mean, let's face it, it's one thing to sit there and say, E. coli's got maybe 250,000 bases, or 750,000 bases, maybe 1 million. Human has got 3 billion bases. That's a quite larger piece. The genes are going to be larger, they're going to be more complex, and so you're going to require larger vectors like viruses. You might use TI plasmids for plants, and therefore you have a bacterial plasmid useful for genetic engineering in plants. You might even use artificial chromosomes, and these are large sort of artificial chromosomes that they built, pieced together pieces of chromosome, so that you put a big long piece of DNA attached into that chromosome and put it into cells. Then you might do something called gene guns. Oh, what? Are you kidding me? This is part of the technology called biolistics, and what happens is this. How do I get it inside the cell? They've been able to take large pieces of DNA, wrap them on little fragments or little pieces of tungsten. Sometimes they use also gold. And they use a gas to shoot them onto a plate, a cultured plate. Now, these particles, either metal or plastic, coated with the DNA, get inside the cells. Once they're inside the cells, they start getting integrated into the cell cultures, cells, DNA. And there you're able to do some really fantastic work, okay? Now, what are some of the concerns about genetic engineering and DNA technologies? It is interesting for you to note that when the human genome experiments and the funding for that research project in the 1980s began, a certain amount of funds for the Human Genome Project was put aside for legal, ethical concepts that had to be explored. So it was not as if the scientists and the legislators wanted to just say, yeah, study it, go willy-nilly, do whatever you need to. They actually said, look, we have to be considering this for what are the privacy issues, what are the health and well-being issues of our patients, what about the perspective of other problems. So the ethics of using genetic information, from genetic information to genetic diagnostics, has raised great concern in the public. Privacy as well as genetically modified organisms are key concerns, as well as clear understanding of the effects of the genes on the health and well-being of the patients. Also, new knowledge of disease genomes develops. And this is where you're starting to get people get very nervous about, well, could this be used for the development of biological weapons or weapons of bioterrorism? Even to this day, I've gone to conferences, and one of the things that they talk about are two areas of major concern. Do it yourself, biotechnology, and it's out there. You can look at it, DIY, and they're doing remarkable stuff. But what if somebody was of a less than sober mindset, or really a bad apple that wanted to make stuff that would be harmful to people? That's a great concern. The second concern is what's called synthetic biology. And this is actually taking pieces of the different genes components and making new life. The potential for that is great in the sense that, hey, I could make organisms that consume pollutants and just release carbon dioxide and water. How wonderful. But what if this was also used to make something else, like a new disease organism? That's where there has been concern and discussion and review, etc. Okay? So we go from here, and we're going to go into a new area, and that's going to be talking about DNA sequencing. Now, DNA sequencing is extremely important because, one, the onset of the Human Genome Project has led us to the development of new techniques and technologies for mass gene sequencing. In other words, we're moving from the point where it might have taken a day to do a small sequence to a couple of hours and you've done the entire human genome. And a lot of this was by the development of robotics, computer data analysis, mathematics and algorithms, as well as the actual biochemistry in doing the genome sequencing technology. Now, the next thing you're seeing is an area called the Human Microbiome Project, and that is applying genomics to the study of the normal microbiota, biological ecosystems that are in the human body, whether it's the gut, whether it's the mouth, whether it's on the skin. These are all very important because we can tell whether somebody, how they are doing with their diet, whether they're susceptible to disease, whether there's new mutations that are leading to new diseases, etc. So what I want to do is explore some of these techniques of basically DNA sequencing. And what's going to happen is we're going to talk about this. Now, if you understand, we start off with some very straightforward techniques. We know that we have an enzyme called DNA polymerase. And if we have free nucleotides, you can use DNA polymerase and one strand of DNA, and then you have to have a primer, which is a piece that kind of attaches to the complementary side of that single strand of DNA. And then what happens is you're going to make that DNA strand with the DNA polymerase. Now, you see the final product, okay? Great. Then we get to the next issue, and that is, and this is all done what they call in vitro, meaning in glass or, in a laboratory test tube, etc. DNA synthesis. Now, this is where, and this is part of the dideoxy chain termination, is also known as chain termination. Remember that we used DNA polymerase, we had a primer, we had free nucleotides. Now, what if we had a nucleotide that was instead slightly different? In other words, it didn't have the 3' OH. So basically, it didn't continue the chain formation. And so chain elongation is terminated. Now, if you mix this up, and I've done this technique, to be honest with you, and use dideoxynucleotides, you can eventually extract from this a process to say, okay, A is here, and then I make another chain, and the A is down further, and that's where it stopped, and then I make another chain, and the A is stopped here. And you can separate all of these chains that have been terminated of various lengths, and you can tell where the A's stop at certain places. So at the 33 mark, at the 37 mark, at the 49 mark, at the 44, the 54 mark, the 56 mark, etc. This is all separated out by gel electrophoresis, mind you. But then just with just A's, you can also do it with the C's, the G's, and the T's. And therefore, you create this large array, as you see here. And you're able to tell in decreasing fragment size what's the actual sequence. Now, what happened was originally, this was a process with 4 tubes. They all have DDNPs, meaning dideoxynucleotides. And you would run this process. Everything would have DNA polymerase. They would have the proper primers, and you'd be able to sequence this whole strand. That's cool. And then you'd separate it out by gel electrophoresis, add a certain dye, and you could tell by the bands. But instead, they began to move to the next step. And the next step was, instead of just having dideoxynucleotide, you put a tag, a fluorescent tag to it. Each color represents a different base. So as you can see in this diagram, greens are A's, reds are T's, blue is C, and yellow is G. And so you'd start to get this sequence that was so cool. Now, I know you're sitting there going, he doesn't have a life. Yeah, I do. But think about it. You've been adding on to this technique. And then you know what the next part is? You have this done by a computer. It will read the different bands of different colors for the different bases, record it, state where the bases are, and you just go from you've gone from basically doing this on a test tube with a small gel electrophoresis unit to a large gel electrophoresis unit to now an optical scanner that identifies the different codes, the different bands, the different colors, and able to, with the computer and the computer algorithm, compile it together so that you have this vast strand that you can tell the C's, the G's, the T's, the A's, and going all the way through. Now, this is where we're going with what is referred to as next-gen sequencing technology. It's all automated. And you combine the DNA sequencing with the capability to align and merge the DNA sequence fragment information. In other words, if you do it with one fragment, you do it with another fragment, you do it with another fragment, they're all going to have a little bit of overlap, so then you're able to piece this together that suddenly, instead of 10,000 base pairs that you haven't gotten an idea, you now have this long map of 10,000 base pairs for a particular protein. And you have the entire sequence of that strand. Now, what do you do with it? Well, there's actually a lot you can do with that. You would be surprised. One of the things that you can't do is you can now compare that between those that are healthy with those that are ill. Now, it also takes a little bit of time because you have to do what's also called, and this is where the computers come in. You notice that you have the data. You notice you got to start aligning it. And you notice that you eventually have the result. Keep reading the textbook. Yes. Okay. Because each one of those strands had different pieces of information. Okay. Keep reading, and then TH. Keep read. And then you had ing the textbook. And you would have the alignment done by the computer, and the end result is that whole statement. Okay? Now, polymerase chain reaction, and I've done this work too. I actually had the good fortune one time I was doing some consulting, and I got to talk with Dr. Kary Mullis. Kary Mullis in 1983 developed it. It made companies billions. It was used all over the world in all sorts of applications, from forensics to genetic diagnostics to identification of ancient dinosaur DNA and so many other things. Here's the idea. I got two choices here. I've got the signal, which is my DNA that I'm interested in, and then I've got my background noise, which is all the rest of the DNA. So it's sort of like the analogy of looking for the haystack, the needle in the haystack. How do I find it? How do I find it? I can't. So I've got a choice. I've got to either make that needle easier to see against all of that haystack, or make lots more needles, or reduce the haystack. Well, in this case, what Kary did was he would increase the signal against the background noise. He would make a lot more copies of the particular DNA of interest. And so eventually you could make literally, in a very short period of time, billions of copies of that particular piece of DNA. And as I've said, it's been used for gene analysis, genetic disease detection, gene replication, evolutionary studies, forensic identification, etc. What's going on? Sorry about that distraction. If you take a look, if you go through page 245 and 246, etc., and there's actually a nice perspective on 247. Here's the deal. What you do is you're going to melt the DNA, and by melting it, you just put a little bit of heat, and that separates out the hydrogen-bonded 2 strands. Then what you're going to do is you're going to anneal or attach a primer. Now, the primers are RNA, but these are used to basically what? Target the start of the DNA polymerase, okay? You also add a lot of free nucleotides so they can basically be incorporated into that new strand, okay, that you're building. As you separate the 2 strands, you attach your primers. And they attach when you started to cool down the DNA. And then what happens is you have a very unique type of DNA polymerase that works at a much higher temperature. This again was a material, an enzyme that was extracted from Thermus aquaticus. This was an archaea type of bacteria found in hot springs. And this DNA polymerase can handle high temperatures without basically denaturing. So you go from cooling it down to 50 degrees Celsius, so the templates anneal. You bring the temperature back up to 72, and you raise this for only a short period of time. Each one of these steps is about 1 minute, folks. 1 minute. When you raise it to 72, boom. The DNA polymerases attach, directed in part by the primers, and they go moving forward, making only the DNA of interest. And after the minute's up, boom, you heat and you separate again. Okay? Now, if you keep going through this cycle, process after process after process, you have ample amount of primers, you have ample amount of basically the free nucleotides. The thermostable DNA polymerase is able to work. What happens? You start with 2, then you get 4, then you get 8, then you get 16. Basically, you're going through a replication process. And basically, the section of DNA that you want continues to mathematically increase. In essence, you make a lot more needles. And what happens in the end is you're able to then extract it all using gel electrophoresis, and you've got this huge whopping band on the gel that basically is just the sequence that you're looking for. How about that? The band of target DNA is going to be very, very clear. You can cut it out of the gel. You can do sequence analysis. You can insert that big whopping amount of DNA into a plasmid, or you can do any other actions you want out of that. And yes, it's relatively easy chemistry to remove the DNA from that agarose gel. Okay? Now let's move to DNA probe technologies because we want to move through the last couple of pieces here. It gets interesting, but I just want to give you some appreciation for it. Remember we talked about earlier signal versus noise. Now, the next techniques are used to locate and magnify the amount of DNA present. The model for the approach, signal versus noise, okay, noise is the undesirable DNA, signal is the DNA of interest, but either we have to use the PCR approach, or we have to hybridize it. We have to find some means to locate that needle in a haystack. That's where we come to nucleic acid hybridization. You have a DNA probe. This is a strand of DNA that contains a sequence complementary to your desired target DNA, and it'll bind to a single-stranded DNA. Now, the high specificity of the base pairing interactions between the 2 strands of DNA, your probe and your target DNA, can be used to locate a specific sequence. So what do? This is where you get into, and you can see where you have the probe, and you see The probe, by the way, has got a little label on it. Now, the label used to be some radioactive isotope. Nowadays, we don't use that at all. We don't want to deal with radioactivity in the lab. And so what happens is most of the time now, you end up with a fluorescent or even a light-emitting label, okay? So you grow colonies on your agar plate, let's say bacteria, and I'm looking for a particular sequence that tells me that this has got a nasty type of toxin that this bacteria produces. What you do is then you have this very interesting material called nylon membrane. The colonies are transferred in place. They're blotted, just like, remember we did the that basic technique with the, sorry about that, that basic technique where we used with the velvet, the replica plating last time. But instead of replica plating, what we're going to do is we're going to take the colonies, we're going to put them onto the nylon membrane. We soak them in an alkaline solution. This will lyse the cells, break open the cells, and it will also denature the DNA. So when we denature the DNA, what have we done? We have basically separated the two strands of DNA by basically taking apart their weak hydrogen bonds. And as a result, now you've got single-stranded DNA open and available for the attachment of the probe. You add the probe, which is a specific nucleotide base. It's got the label on it. And then you allow for hybridization, okay? And the regions of DNA complementary, they will hybridize with each other, okay? And what happens there? Boom, you've got labeled DNA. Now, with colony blots, the method of identification of DNA of interest is in specific colonies, and you can then tell which colonies have the clone DNA of interest if you're looking for DNA clones, okay? Now, southern blot, and yes, there's a different types of blotting. One of them is called southern. They actually have one called northern, which is for RNA. Southern blot. You'll see this on 920, but wait a minute. Let me go back here. The separation process of restriction fragments, separated by the length of fragment that use gel electrophoresis to separate out the DNA fragments. Fragments are then transferred to the membrane filter and hybridized with a DNA probe for identification. That's the southern blotting approach. In other words, if I'm looking for a DNA, rather than go through this process with hybridization of bacteria cultures, I might take a large amount of DNA, chop it up with restriction enzymes, run it through the gel electrophoresis, transfer the DNA from the gel electrophoresis onto the nylon membrane, and do the hybridization there. Still, what's the key point? The DNA probe. Now, gel electrophoresis, I've mentioned it to you before, major technique, used to separate out the restriction fragments. One thing to keep in mind when you're doing this, and I'm bouncing around here, so please bear with me. You can get similar strands, and you can use restriction enzymes, and the restriction enzymes will cut up different pieces of DNA. Now, if let's say there's a particular piece that can't be cut by that restriction enzyme because there's a slight mutation, a one-base change in the cut area. That means instead of two smaller fragments, you get a larger fragment. This is known as restriction fragment length polymorphism, RFLPs. This is the process where basically it's very sensitive. It tells you if there's single base differences at the restriction sites. And you can isolate the bands, and you can use them for all sorts of identification, from single base pair mutations to genetic fingerprinting of humans, as well as determination of genetic mutations within humans or other species. Now, let me turn you back to fluorescent in situ hybridization. Remember we talked about making a hybrid by basically using a gene probe? Yeah. You had that right here. Okay. Well, what happens if I use intact cells and I just basically have cells that are attached to a microscopic slide? Could you do that gene probe routine as opposed to having to take all these cells and put them onto a nylon membrane? Yes, I can. The in situ means in site. So what you're talking about is you fix the cells to the site, you rupture the cells, the DNA is denatured on the slide, you add the probe, you allow it to anneal, you wash off the excess probe, and the detection of the probe occurs. And what you've got here are a series of different probes in this photo. This is 921 in your text. And what they're looking at is the actual microbiota inside of an earthworm gut. In other words, you've got a variety of bacteria here, and you use different probes, and the different colored probes will identify the different types of bacteria present in that gut. That is a really high-powered, speedy process, and that's what I'm trying to get you to appreciate. Finally, the last one is DNA microarrays. In what we found we've been able to do, and by the way, there's been a lot of discussion for the past 20 years using microarrays as being a chip that you would do diagnostic, and guess what, folks? Yeah, it would be coming back to you. You might be able to say, could I have a drop of your blood? You put it on the chip, you just put it into the reader right next to the patient's bed, and you'll be able to get information from that. What you have is a known mix of different sequences for a specific gene, and they're placed to hybridize with an unknown DNA. And so the degree and occurrence of hybridization of the complementary sequences can determine the level of gene expression as well as the presence of select genes of interest. So what you see on this particular slide, think about this. The really brightly lit up areas are saying this sequence is present. The dark areas are saying that there's not that sequence present. And that's the key. I want you to have an appreciation for this. You're not expected to do it, actually, but don't panic. You may be seeing this coming down the road. As we get further and further with microarrays and everything, the price drops down, the techniques get better and more sensitive. It's conceivable that someone, and it sounds almost like Star Trek, will eventually be able to say, take saliva, take blood or whatever, and put a drop on a slide, on a microarray, slip it into a reader, and it may be a reader that'll be right next to the bed of the person, and they'll be able to monitor the status or the presence of certain microorganisms there. Well, we're just about done, so this is what I would say to all of you. Review the microassessment, review the chapter summary, and we will move into the next section. And if I'm not mistaken, we will be coming up to an exam. Please read the instructions on the exam carefully. In week 5, your first exam will cover only weeks 1 through 4 for the content, okay? You will have some reading that you have to do, but that's going to be for the next week's content. And I hope everyone has a great week.